Hello guys and girls, myself Dr. Omkar Sangita Dilipra Sonone, and today I welcome you all again to the session of Sparks, okay, where we will be discussing about the high yielding topic from pathology. And the most important topic from pathology that we need to discuss over here is nothing else but your cell injury and cellular adaptations. This topic has come multiple times in the examinations. There have been questions in and around this topic, so please remember, always focus on this topic as this is very much important. Let's begin the session. First of all, I would like you to go through this quote, that is, stop doubting yourself, work hard and make it happen. Nothing is impossible, okay? So please remember, hard work is the key. You need to work hard, you need to revise all that stuff that you have, and just you will be on the other side of the fence along with us after the December examination. So let's begin the session without any further delay. And the topic we'll be going to study is your cell injury, okay? So please remember, as we all know, cell is the basic anatomical and functional unit of life. So whenever there is some stress or there is some injurious stimuli on the cell, it would lead to some different changes in the cell, right? So we will be discussing about those changes, okay? So please remember, whenever there is some stress or some injurious stimuli, it would lead to some changes in the cell, and these changes would manifest differently. So these changes will be we are going to see today. So the important one is, when the cell is under stress, there would be some changes in different organelles also. And which are the most important organelles which will undergo changes? So please remember, first of all, what is the most important organ? That is our brain of the cell, that is nucleus. So please remember, nucleus would be affected. Second, the powerhouse of the cell, that is your mitochondria. Next one, it's your endoplasmic reticulum, factory of the cell. And last one is the protective layer of the cell, that is your plasma membrane. So these are the four most important organelles which are affected during cell injury: nucleus, mitochondria, endoplasmic reticulum and plasma membrane. Now first of all, let's discuss about the causes. Okay, there can be multiple and different causes of cell injury, but the most common cause on which questions have been asked multiple times. So please remember, the most common cause of cell injury is nothing else but hypoxia. So please remember, the most common cause of cell injury is hypoxia, that is decreased oxygen supply to the cell. So the question can be modified a bit. So they can ask you, the most common cause of hypoxia is what? So please remember, if the question is most common cause of hypoxia, then your answer should be ischemia. So please remember, the most common cause of hypoxia would be ischemia. And what is the difference between hypoxia and ischemia? Hypoxia is decreased oxygen supply to the cell, whereas ischemia is decreased blood supply to the cell. Okay, so most common ones are these ones. इसके अलावा there can be different causes of cell injury. There can be some physical agents, there can be some chemical agents, okay, immunological, genetic, infectious, nutritional agents. All of these are responsible for causing cell injury, okay? So but remember, the most important ones to be remembered are the most common cause, hypoxia, and most common cause of hypoxia, ischemia. Okay, so the questions can also come like, the cells which are most sensitive, to hypoxia. So please remember, the cells which are most sensitive to hypoxia are your neurons. So please remember, neurons are the cells which are most sensitive to hypoxia. Okay, degeneration of neurons starts within minutes of hypoxia. Okay, that is very much important. Next, they can also ask you cells which are least sensitive to hypoxia, and then your answer should be fibroblasts. So please remember, the cells which are least sensitive to hypoxia are fibroblasts. If fibroblast is not an option, the next option would be skeletal muscle. So please remember, cells most sensitive to hypoxia are neurons. Cells least sensitive to hypoxia are fibroblasts, followed by skeletal muscles. Now, if there is some injurious stimulus onto the cell, there can be different manifestations of the cell. Whenever you are under stress, okay, like for example, you have started your FMG preparations, you have come to Gautam, Nagar, there is a lot of stress. So what is the first thing that you try to do? You try to adapt yourself to the new environment, and that is what the cell does also. Okay, so please remember, the cell also tries to adapt to the newer environment, the stress. Okay, so the first change that would occur is your cellular adaptations. There are different types of adaptations that are seen in the cell. So please remember, the first change is cellular adaptation. If the injurious stimuli still persist, okay, then the cell will undergo reversible cell injury. Then the cell would undergo reversible cell injury. And what do we mean by reversible cell injury? That is, as soon as we withdraw or take off the injurious stimuli or the stress, the cell will restore its normal physiological state. Okay, the cell will restore its normal functioning state. Okay, that is what we mean by reversible cell injury. If the injurious stimuli still persist, if the stress is still there, then the cell would undergo irreversible cell injury. And what will happen in irreversible cell injury? In irreversible cell injury, whenever the injurious stimuli or stress is withdrawn, then also the cell could not restore its normal functioning state. Okay, that is what we mean by irreversible cell injury. Irreversible cell injury can further progress to cell death. It can further progress to cell death. Cell death can be carried out by different mechanisms. Okay, before discussing cell death, we'll discuss other two phenomena which can be seen in cases of cell injury. Whenever there is stress, there can be some intracellular accumulations. There can be some pigment which can accumulate inside the cells, and that can lead to intracellular accumulations. And the last one, there can be pathological calcifications. There can be some pathologic calcifications. Okay, so now let's discuss about the cell death. There are 5 different mechanisms of cell death. So just remember the names over here. We'll be discussing about cell death in next video. So please remember the important ones are your apoptosis, which is programmed cell death. Next one is your necrosis. Then the next one is necroptosis. As you can see, there would be combination. Then next one is your pyroptosis. And the last one, which is recently added, is your ferroptosis. नाम से पता चल रहा है that it is a iron mediated cell death. Okay, so important 5 things about cell death. The 5 mechanisms of cell death are apoptosis, necrosis, necroptosis, pyroptosis and ferroptosis. Now let's discuss further. Okay, as we have discussed that the first change which is seen in cells after injurious stimuli is applied to them, that is cellular adaptations. And there are 4 different kinds of cellular adaptations which are seen. The first one is your hypertrophy. The next one is your hyperplasia. Third one is your atrophy, and the last one is your metaplasia. These are the 4 different kinds of cellular adaptations. So we'll start discussing with hypertrophy. So what do we mean by hypertrophy? Please remember, hypertrophy is increase in cell size. Hypertrophy is increase in the cell size and not increase in the cell number. Not increase in cell number. So please remember, cell size to increase ho raha hai, but cell number is not increasing. And as the cell size increases, there is also increase in organ size. Increased organ size is seen. So what is the cause? What is the mechanism of hypertrophy? So please remember, the mechanism of hypertrophy is increased protein synthesis. Okay, mechanism of hypertrophy is increased protein synthesis that leads to increase in the cell size. So there is only increase in cell size but not increase in the cell number. That is, it occurs only in non-dividing cells. It occurs only in non-dividing, also known as permanent cells. It occurs in non-dividing or permanent cells. Important one to be remembered. Moving further, guys, now to our examples of hypertrophy. And what are the examples of hypertrophy that we need to remember? Please remember, there can be some physiological examples and there can be some pathological examples of hypertrophy. So let's begin with the question which has been asked multiple times. That is, the physiological example of hypertrophy is your uterus during pregnancy. So as we all know, normal uterus is around 70 grams, but during pregnancy it almost becomes around 1100 grams. So this is a classical example of uterine hypertrophy during pregnancy. The next example is your breast. So please remember, breast during lactation is an example of hypertrophy. As we all know, there are questions which also tells us about hyperplasia. So please remember, I'll be telling you about the difference. Breast shows 2 phenomena. It also shows hypertrophy during lactation and it also shows hyperplasia during 2 other conditions that we'll discuss now. Okay, and the third example of physiological hypertrophy is your skeletal muscle hypertrophy, which occurs due to strenuous exercise in bodybuilders. Please remember. Now, the next classical example of hypertrophy, pathological hypertrophy, is nothing else but your left ventricular hypertrophy, LVH. Left ventricular hypertrophy is seen in conditions like hypertension or any kind of valvular diseases. Okay, so please remember this. Moving on to our next cellular adaptation, and that is your hyperplasia. So what happens in hyperplasia? There is increase in cell number. So please remember, hyperplasia mein there is increase in cell number, and this is due to the The mechanism for hyperplasia is what? The mechanism for hyperplasia is growth factor induced proliferation. Okay, so proliferation हो रहा है, मतलब cell division हो रहा है. So please remember, in hyperplasia there is increase in cell number due to growth factor induced proliferation, and proliferation is only possible in which kind of cells? It is possible in dividing cells. Okay, it is possible in dividing cells. So please remember, hyperplasia occurs in dividing cells, the mechanism being growth factor induced proliferation. Okay, so now let's see the examples. Okay, there are different kinds of examples. Again, physiological and pathological hyperplasia can be there. So please remember, physiological hyperplasia would be seen in conditions like, there are different kinds of hyperplasia. There can be compensatory hyperplasia. So please remember, compensatory hyperplasia would be seen in cases of hepatectomy. Okay, whenever a part of liver is removed, and as we all know, liver has the ability to regenerate. Okay, the hepatocytes have the ability to regenerate. Therefore, after hepatectomy, the liver would regenerate. It would lead to hyperplasia that cause liver regeneration. Okay, so this is an example of compensatory physiological hyperplasia. The next example that you need to remember is a hormonal hyperplasia. Hormonal hyperplasia, whenever there is some hormonal stimuli onto the breast, it leads to hyperplasia of the breast. Okay, it leads to hyperplasia of the breast, and this occurs during puberty. Okay, please remember, this occurs during puberty and pregnancy. So please remember, now you know the difference. Breast during lactation is an example of hypertrophy, whereas breast during puberty and pregnancy is an example of hyperplasia. Please remember this. Whereas the other things you need to remember about pathological hyperplasia, please remember, pathological hyperplasia can be due to influence of different hormones. Okay, if there is excess of estrogen, okay, excess of estrogen would lead to constant stimuli on the endometrium, and that can lead to endometrial hyperplasia. That can lead to endometrial hyperplasia, which can be a predisposing factor for endometrial cancer. And the next one, increased androgens, increased testosterone can cause constant stimuli on the prostate, leading to benign prostatic hyperplasia seen usually in elderly males. Right? So please remember, these are the examples of hyperplasia. Moving further to our third kind of cellular adaptation, and that is your atrophy. So what is atrophy? Atrophy is decrease in cell size and number both. Okay, so please remember, atrophy is decrease in cell size or number that leads to decrease in organ size also. That leads to decrease in organ size also. So organ shrinks, right? And what is the mechanism? So please remember, the mechanism is decreased protein synthesis. Okay, hypertrophy में क्या हो रहा था? There was increased protein synthesis. So opposite would occur in atrophy, that is decreased protein synthesis would be there, or there was increased protein degradation that occurs. Increased protein degradation that occurs. Okay, so important ones to be remembered. Okay, now let's see the examples of atrophy. The examples of atrophy can be different. It can be physiological again, or it can be pathological. What is the physiological example of atrophy? So during organogenesis, okay, so please remember, during organogenesis many of the organs atrophy. Apart from that, in life there can be thymus atrophy, as we all know. Okay, there can be thymus atrophy or notochord atrophy, and both of these are examples of physiological atrophy. Whereas there are different kinds of pathological atrophy which is seen. First one is your senile atrophy, and senile means old age. Okay, so in old age what happens? There is atrophy of different organs, like atrophy of breast tissue, atrophy of many other organs also occurs during old age. Okay, a classical example of senile atrophy that I can give, a pathological one, that is your Alzheimer's disease. What happens in Alzheimer's disease? As we all know, there is widening of the gyri and sulci of the brain, and that happens due to the brain atrophy which occurs in Alzheimer's disease. That is a classical example of senile atrophy. The other examples would be pressure atrophy can be there. There can be disuse atrophy. Disuse atrophy is important. It occurs in a limb muscles after prolonged immobilization of that particular limb. Okay. Then there can be denervation atrophy. Denervation atrophy is due to the nerve supply which is cut down. Okay, denervation atrophy can be there. There can be nutritional atrophy, which is seen in cases of kwashiorkor, you can say. Okay, so please remember, all of these are different kinds of pathological atrophy which can be seen. Now coming to the last one, the last cellular adaptation that we need to discuss, and that is metaplasia. Please remember, that is metaplasia. So what is metaplasia? Important one. Questions have been asked on the definition of metaplasia even. Okay, so please remember, metaplasia is first of all, the most important word to be remembered is it is a reversible change. So please remember, all cellular adaptations are reversible. All cellular adaptations are reversible. That is, as soon as the injurious stimuli would be removed, the cell would restore its normal functioning state. Okay, so please remember, metaplasia is also reversible. We tend to forget that, we mark it as irreversible, but no, metaplasia is a reversible change in which what happens, in which one mature or a differentiated cell type, okay, one mature cell type is changed to another mature cell type. Okay, another mature cell type. Okay, we can say one mature type of epithelium like columnar epithelium is changed to squamous epithelium. That is a classical example of metaplasia. So guys, you can you tell me, please remember, the important one over here is the most common type of metaplasia. The questions have been asked repeatedly on this, the most common type of metaplasia. So please remember, you need to remember this, the most common type of metaplasia is seen usually in smokers. So what happens in smokers? Usually in the respiratory tract, what is the normal epithelial lining? In respiratory tract, the normal epithelial lining is pseudo-stratified ciliated columnar epithelium. Okay, due to prolonged smoking, due to constant smoking, okay, it is an injury, right? Smoking is an injury to the cell. Therefore, due to constant smoking, this pseudo-stratified columnar epithelium of the respiratory tract is changed to stratified squamous epithelium. Okay, it is changed to stratified squamous epithelium, and this is a classical example of squamous metaplasia. So please remember, what is the most common type of metaplasia? It is your squamous metaplasia seen in smokers. What is the most common type of metaplasia? Squamous metaplasia seen in smokers. Okay. The other example, which is a classical one, that is your Barrett's esophagus. Okay, the other example is your Barrett's esophagus. And what happens in Barrett's esophagus? There is a predisposing factor, which is your GERD, that is your gastroesophageal reflux disease. What happens? Gastroesophageal reflux disease means there is a constant exposure of the esophageal epithelium to the gastric acid. Okay, the HCl keeps on regurgitating into your esophagus, which leads to a injurious stimuli onto your esophageal epithelium. And normal esophageal epithelium is your squamous epithelium. Okay, normal esophageal epithelium is your squamous epithelium, but due to this constant exposure to acid, it changes to columnar. So please remember, what happens in Barrett's esophagus? There is metaplasia from squamous to columnar. So please remember, there is columnar metaplasia in cases of Barrett's esophagus. But if they ask that what is the metaplasia seen in cases of Barrett's esophagus, and the options are squamous, columnar, mesenchymal, and the last one is intestinal metaplasia. So please remember, the answer to be marked is your intestinal metaplasia. That would be your best answer. Okay, so don't mark it as columnar, then mark it as intestinal metaplasia. Why intestinal metaplasia? Please remember, in cases of Barrett's esophagus, intestinal metaplasia is classically seen because there are columnar epithelium is seen, but along with that, one variety of cell is seen, and that is pathognomic of Barrett's esophagus, that is your goblet cells. Goblet cells are mucin secreting cells, and these are characteristically seen in the intestine, but they are also seen in cases of Barrett's esophagus after metaplasia. Therefore, this is a classical example of intestinal metaplasia along with goblet cells. And what is the most pathognomic feature of Barrett's? That is your goblet cells. Okay. Then, talking about the last one, that is your mesenchymal metaplasia. And mesenchymal is your connective tissue metaplasia. And what is the classical example of mesenchymal metaplasia? Please remember, it is your nothing else but myositis ossificans. Okay. So, myositis ossificans में क्या होता है? Due to rubbing, okay, due to rubbing after a fracture, there is constant, if you keep on rubbing the muscles, that leads to ossification into the muscle. Okay, so please remember, there is bone formation inside the muscles, and that is a classical example of mesenchymal metaplasia, myositis ossificans. Now guys, moving further to the next topic that we will cover is our cell injury. Okay, and it is very much important now to remember about cell injury, guys. So let's see. The next topic is, first of all, we'll talk about is cell injury, that is reversible cell injury. Okay, the first one that we'll talk about is reversible cell injury, that I have already discussed with you, that in reversible cell injury, as soon as the stimulus is withdrawn, the cell will restore its normal function. Okay, so let's see the mechanism of cell injury. There can be damage to multiple organs. Okay, there can be damage to the multiple organelles into the cell, but the most common organelle which is affected, this question have been asked previously, the most common organelle which is affected during reversible cell injury is your mitochondria. So please remember the answer to this question, that is the most common organelle affected during cell injury is mitochondria. And what is the earliest change seen? So please remember, what is the earliest change seen? And the answer should be hydropic change. Okay, what is the earliest change seen in a cell after reversible cell injury? It is your hydropic change, also known as cellular swelling, also known as cloudy change. Okay, so please remember what happens in hydropic change or cellular swelling. There is imbibition of water into the cell. Okay, there is accumulation of water into the cell that leads to cellular swelling, and this happens due to, as we all have discussed, the plasma membrane is affected, right? So what is located on the plasma membrane? That is your sodium potassium ATPase pumps. Okay, as the sodium potassium ATPase pumps would be affected, that leads to impairment of their function, and that causes accumulation of sodium inside the cell. That causes accumulation of sodium inside the cell. And as I've always said, wherever there is sodium, water follows. Okay, जहाँ जहाँ sodium है उसका प्यार पीछे आ जाता है, that is water. Okay, so please remember, excess of sodium inside the cell would also attract water inside the cell. That leads to imbibition of water inside the cell, and that leads to this hydropic change or cellular swelling. Okay, the cell would grow so much big in size. Okay, and there would be bleb formation. Okay, so cell would have blebs on it. Okay, cell would have blebs on it, and this is the earliest change and also the most characteristic change of reversible cell injury, that is hydropic change or cellular swelling, also known as cloudy change. Along with it, there can also be, as plasma membrane, there can also be different enzymes which are activated. Okay, and these different enzymes are your breakdown enzymes, like your phospholipase enzyme is activated. Okay, and as soon as this phospholipase enzyme is activated, it leads to breakdown of phospholipids, and phospholipids, they make up your cell membrane. So as soon as this phospholipase enzyme is activated, phospholipids are broken down, and that leads to a characteristic appearance, that is your myelin figures. It leads to myelin figures. What are myelin figures? You can see them like this. Okay, these are concentric rings of the broken down phospholipids. Okay, those are known as myelin figures, which are also seen in cases of reversible cell injury. Okay, apart from that, now we have discussed about plasma membrane, we have discussed about mitochondria. So what is the change which occurs in the third organelle which is affected? Okay, so please remember, the third one which is affected is your nucleus. And what happens in the nucleus? Nucleus में क्या हो जाता है? So please remember, as in nucleus there is increased anaerobic glycolysis. Okay, there is increased anaerobic glycolysis in the cell due to decreased oxygen. As we have discussed already, hypoxia is the most common cause, मतलब oxygen कम है. That leads to increased anaerobic glycolysis. And anaerobic glycolysis का last product क्या है? The final product is your lactate, which causes lactic acidosis. Excess of lactate would lead to lactic acidosis, and finally lactic acidosis would further lead the cell to decreased pH, and this decreased pH in the nucleus can lead to clumping of chromatin. Okay, there can be clumping of chromatin inside the nucleus. So please remember, what is the nuclear change which is seen in reversible cell injury? That is your clumping of chromatin. Now the last organelle which is left is our endoplasmic reticulum. And what is the change seen in endoplasmic reticulum? As we all know, water is imbibed into the cell. Okay, there is cellular swelling, so therefore there would also be swelling of your endoplasmic reticulum, and this leads to the detachment of the ribosomes. As we all know, rough endoplasmic reticulum has ribosomes attached all over it. So as soon as the endoplasmic reticulum swells out, there would be detachment of these ribosomes, and ribosomes का function क्या होता है? They cause protein synthesis. As soon as there's detachment of ribosomes, there would be decreased protein synthesis. Okay, so please remember, what are the different changes seen in cases of reversible cell injury? In plasma membrane, impairment of sodium potassium ATPase pump leading to cellular swelling or hydropic change, which is the earliest change seen. Okay, and then there would be also formation of myelin figures. There would be endoplasmic reticulum में there would be decreased protein synthesis. Nucleus में there is clumping of chromatin which is seen. But as we all know, what is the most characteristic feature of reversible cell injury? We have discussed it. It is your cloudy swelling or hydropic change. Okay, so please remember, this should be your answer. Moving on to our next one, guys. The next one would be our irreversible cell injury. Okay, irreversible cell injury I've already discussed. What is irreversible cell injury? That is, as soon as the injurious stimuli is withdrawn, okay, the cell would restore its normal physiological function. The cell would restore its normal physiological function. Okay, important. Now let's see what happens in irreversible cell injury. First of all, there is plasma membrane dysfunction, and we have already discussed about this. As there is plasma membrane dysfunction, the sodium potassium ATPase pump is gone, therefore there is cellular swelling. Okay, but now there would also be loss of selective permeability. There would also be a loss of selective permeability. Okay, as we all know, the plasma membrane is selectively permeable. It allows some substances, it refuses some substances, but as soon as there is dysfunction, it loses its selective permeability, which causes increased influx of calcium inside the cell, and there would be increased cytosolic calcium. Okay, there would be excess of calcium which is accumulated into the cytoplasm of the cell, and as we all know, calcium is a stimulus for the enzymes to be activated, and therefore calcium activates different kinds of enzymes. Okay, calcium activates different kinds of enzymes. Please remember this. And calcium activates, as we all have discussed, first of all, it activates your phospholipase enzymes, which breaks down your phospholipids of different cellular or organellar membrane, and that leads to the formation of what? That leads to the formation of myelin figures. Okay, so there would be membrane damage, and that caused the myelin figures to be formed. Then the other enzymes which are activated are your proteases, which will break down different kinds of proteins. The other enzyme which is activated is your endonuclease, that will break down your DNA. Okay, that breaks down your DNA. And the last enzyme which is activated is your RNase, that would break down your RNA. So please remember, okay, as soon as the selective permeability of the plasma membrane is lost, there is excess of cytosolic calcium accumulated in the cell, which leads to the formation of myelin figures, decreased protein synthesis, DNA degradation, and RNA degradation. Okay, and as the mitochondrial damage occurs, as we all know, mitochondria is the powerhouse of the cell, that leads to decreased ATP synthesis. As soon as mitochondrial damage occurs, there is decreased ATP synthesis which is seen. Okay, and it also leads to the formation of large flocculent amorphous densities inside the mitochondria. So please remember, large flocculent amorphous densities in mitochondria are seen in cases of irreversible cell injury. They are seen in cases of irreversible cell injury. Important one. Okay, whereas now we'll see the changes in nucleus. In cases of irreversible cell injury, the different features which are seen are your pycnosis. The second one is your karyorrhexis. Okay, the next one is your karyorrhexis, and the last one is your karyolysis. So please remember, pycnosis में क्या होता है? There is clumping of chromatin. Okay, first of all, the first feature that would occur in nucleus is your clumping of chromatin. That is known as your pycnosis. Then karyo means nucleus, rhexis means fragmentation. So what happens in karyorrhexis? It is your fragmentation of your chromatin. So please remember, karyorrhexis is fragmentation of your nuclear chromatin. Okay, and lastly, karyolysis means, karyo means nucleus, lysis means dissolution. So please remember, lastly what will happen? There would be dissolution of all this nuclear chromatin. Dissolution of nuclear chromatin. So please remember the three features that are seen in cases of irreversible cell injury sequentially. First is pycnosis, clumping of chromatin. Second is karyorrhexis, fragmentation of nuclear chromatin. And last is karyolysis, dissolution of nuclear chromatin. Please remember, whereas the most characteristic feature, please remember, the most characteristic feature which are seen in cases of irreversible cell injury is nothing but your nuclear changes. Please remember, the nuclear changes are the most characteristic features which are seen in cases of irreversible cell injury, because it helps us to differentiate it from the reversible cell injury. Reversible cell injury में only क्या हो रहा था? There was just clumping of chromatin. Okay, so please remember, these are the features that you need to remember about irreversible cell injury. Okay, so just before ending the session, I would like to just revise it in a nutshell. Reversible cell injury and irreversible cell injury, what are the differences? Okay, reversible cell injury, the most common organelle affected is your mitochondria. The earliest change seen is your hydropic change or cellular swelling. Okay, and the other changes which would be seen in plasma membrane, there would be Impairment of the sodium potassium ATPase pump, leading to imbibition of water, leading to hydropic change or cellular swelling. It also leads to bleb formation onto the cell. Then endoplasmic reticulum would undergo swelling, leading to decreased protein synthesis. Nucleus mein there is excess of anaerobic glycolysis, leading to accumulation of lactic acid, finally decreasing the pH, leading to clumping of chromatin. And the most characteristic feature of irreversible cell injury is cloudy swelling. Whereas irreversible cell injury, there would be plasma membrane dysfunction, there would be loss of selective permeability, there would be increased cytosolic calcium, activation of different kinds of enzymes, myelin figure formation, and degradation of proteins, RNA and DNA. Whereas mitochondrial damage would lead to decreased ATP synthesis, and there would be characteristic large flocculent amorphous densities. Okay, so please remember, large flocculent amorphous densities were not seen in cases of reversible cell injury, they are only seen in cases of irreversible cell injury. Whereas decreased ATP synthesis is seen in both, reversible में भी होगा, irreversible में भी होगा. Whereas in nucleus there would be different kinds of changes like pycnosis, karyorrhexis, karyolysis. And the most characteristic feature of irreversible cell injury are your nuclear changes. Okay, so with this we come to the end of this session, and I hope this session might have helped you, and we'll soon meet in the next session of Sparks, okay, with some other high yielding topic and also with some other MCQ discussion. Thank you so much guys, keep working hard, and let's meet soon.